Hello, I hope you're doing well. This video today is going to be about mitosis. And mitosis is the process of replicating the for a eukaryotic organism. So mitosis stands for cell replication for eukaryotes. Okay, so for prokaryotic cells, there is a different process, and that is called binary fission. But for this class, we are only going to focus on mitosis, and that is when a cell splits into two cells. And this process is a little bit complicated, and this is specific to eukaryotic organisms, which includes our cells. Because we're eukaryotes, our cells undergo this kind of process. Before I get into all the details, there's a little bit of background that I need to cover first. First, I want to talk about what the chromosomes or what the DNA looks like inside the nucleus. So let me just draw a little picture of a cell. And here is the nucleus. Okay, and so far we've always been talking about DNA is being inside the nucleus. Okay, sorry. And the DNA looks different depending on the organism. I mean, it's different based on how the amount. Okay, it looks exactly the same. It's a double helix for all living organisms, but the amount of DNA is different based on the different organisms. And I'm just going to focus on humans because we're human and it's just easier to just have an example. And so humans have DNA in chromosomes like all organisms do, and humans have 46 chromosomes. Okay, and we have 46, and all eukaryotic organisms have chromosomes in pairs. Okay, and so it's always going to be an even number for a eukaryotic organism. Our number is 46, and the chromosomes come in pairs because one chromosome comes from your mother and one chromosome comes from your father. And so you have to have them in pairs. And what I'm going to do is I'm going to show you a picture of the human chromosomes. So what this picture is called a karyotype. Let me find my karyotype here. Here it is. And so what we do as humans is we like to categorize everything. And so let me just scoot over here so we can look at it. So this is called a karyotype. Let me just write that word down. And a karyotype is just all the organisms, all the chromosomes lined up in order. And so what we do as humans is we make the biggest one number one, and then the smallest one the last pair, okay? And so this is a human karyotype. And so what this is showing is that each chromosome type, we have two. So for chromosome 1, we've got one chromosome from mom, and then one chromosome from dad. For chromosome 2, we've got one from mom, one from dad. For 3, we've got one from mom, one from dad, and so on. So all the way to 23. 22 is the smallest chromosome. And our 23rd pair is called our sex chromosomes. I'm going to write that down. Sex chromosomes. And the sex chromosomes come into two types. There's either an X or a Y. And so if you are XX, then you are a female. If you are XY, then you are a male. Okay? And so this karyotype has an X and a Y, so this is a karyotype for a male. Okay, and all the chromosomes have genes on them, genes being a section of the DNA. And so I'm pointing this out to you because all this DNA is going to double before the cell can replicate, okay? And so I just want to point out what the chromosomes look like to you. Okay. And so this is what I'm going to tell you is that, okay, so back up just a second. I'd like to just review the cell cycle before I get into the details of mitosis. And so the cell cycle was in the last video, but let's just review it real quickly. So the cell cycle involves two major steps. Okay, it involves the first step being interphase, and the second step is mitosis, which we're going to talk about today. So step one is interphase. And interphase is broken down into three steps. We have G1, where the cells live in its life. I'm just going to living. The S phase is where all this DNA gets replicated. Okay, so DNA is doubled. I'm going to put replicated because we know about the DNA replication process. And so what happens is a chromosome, like we saw in the karyotype, originally looks like this. So let's just say this is chromosome number 1. Here's mom's and here's dad's. Okay, they have the same genes on them. They just might have different versions of that gene. For example, with eye color, maybe your mom has brown eyes, your dad has blue eyes. Your mom's chromosome might have the brown eye gene, but your dad's chromosome might have the blue eye gene. But they still have the eye color gene. So all the genes are the same. It's just they might have different versions. However, during the S phase of interphase, these chromosomes will completely replicate. So we will make a whole copy of them. So after DNA replication, so this process here is DNA replication. This is what they're going to look like. This chromosome of mom's is going to just copy into this. Okay, sorry about my drawings, they're not that great. And then dad's chromosome is going to copy like this. Okay, and so a couple of things I want to point out to you here is that the two chromosomes are held together by a belt called a centromere. Okay, centromere is the belt. And each of these chromosomes are identical. And they are called sister chromatids. Sister chromatids are identical. Okay, so this is dad's chromosomes, and it replicated. And it also has a centromere held together. It's like a little belt, holds the two twins together. And these are also called sister chromatids. They're identical. Okay, last thing, on the centromere, there is this little bulge of a protein. I'm just going to draw a little circle here. This little protein bulge is called a kinetochore. Okay, this is it's like a handle. It's like, I like to think of it like a love handle, like when you wear your belt too tight and your waist kind of goes over the belt, and that's your love handles, right? And so this is called a kinetochore. It's the love handle. Okay, that's going to come into play during mitosis. So you need to know what that is. Okay, so we're in interphase. This is cell cycle. This is the life cycle of the cell. G1 is it's living. Now it's got to replicate. So it's going to go into S phase. The DNA has all replicated. So in this karyotype up here, each of these chromosomes are going to replicate, but they're going to stick together, and they're going to be sister chromatids. Okay. The second thing that happens in the S phase is this other structure replicates called a centrosome. Okay, so I'm just going to put, I guess I'm going to circle this. So DNA is replicated. And the second thing is that the centrosome is replicated. Oops, is replicated. And this is this, I showed it to you in the last video. It's these little barrels. They look like little barrels, and they are necessary for mitosis. So they replicate in the S phase. I'm going to pull in a picture to show you what they look like. In the last video, I drew them for you, but here I'm just going to let you see what they look like in a cartoon picture from your textbook. Okay, so that's very large. Let me shrink that up. Okay, so here is what a centrosome looks like. Right here on the left is a picture of the cell. And then here, this big purple thing is the nucleus. Inside the nucleus is the DNA. All the DNA has replicated. Okay, they've turned into from one chromosome to sister chromatids. And then outside the nucleus, there's this thing. This is called a centrosome. And it's just like these two barrels. To me, they look like barrels, and they're crisscrossed. Okay, so this is what it looks like. Okay, each one is called a centriole. Together, they're called a centrosome. But the centrosome is going to replicate. So you have one in every cell. When it's in S phase of interphase, it's going to become two. Okay, then the cell goes into the next phase, which is G2. So let me say step 3. Let me just scoot up here real quick. I just want to make sure I numbered it. Okay, so we have, interphase is G1, S, and G2. Oops, I'm sorry. So I'm not numbering it, sorry. Trying to keep my outline consistent. Oh, okay. So the next phase is G2, and this is a checkpoint. So what the cell is going to do at this point is check all the DNA, make sure there's not too many mutations. If there are too many mutations, the cell is going to kill itself, and then that's it. It's over. Okay, if everything looks good at G2, it's going to move on to the next phase, which is what we're talking about in this video, which is mitosis. Mitosis is a lot of different steps with some major processes happening in each step. Okay, so what I'm going to do is I'm going to list I'm going to write the name of the step, tell you the highlights of that step, and then draw it. Okay, so you're going to need room in your notebook. Okay, there are 5 steps in mitosis. So I'm going to color code these. I'm going to make them, maybe I'll make them orange. Yeah, orange. Yeah. First step is called prophase. I'm just going to capitalize it. Prophase is the first step. And so remember, we have a cell. I'm going to draw the cell. And oops. Oh my goodness, what's happening here? Not right now. Okay. Oh my goodness. Okay, I'm going to draw a cell. So we're going to draw a cell for each stage. And right now, the cell has a nucleus and it has all the chromosomes in here. I'm going to draw 2 pairs. Okay, so this is one pair and this is another pair. So we have 23 pairs, but here I'm drawing 2 pairs. Okay, so this is 2 pairs and each one has replicated. So there are 4 chromosomes in this nucleus in my drawing, okay? And so the way that I've drawn them is that they're already replicated. So there's a centrosome holding these together. Okay, let's say this is chromosome number 1, this is chromosome number 2. Okay, so the longest one and the next longest one. Okay, in prophase, what happens is the nucleus falls apart. Okay, so what happens is you have this nuclear envelope and it just degrades. So I'm going to say the nucleus falls apart. Okay, and then the centrosomes start to move apart from each other. So let's say that one centrosome is here and another centrosome is here. They're going to start to move away from each other, okay, because they want to get to the opposite sides of the cell. So the nucleus falls apart, centrosomes move apart from each other. Okay, that's what happens in prophase. And so to draw this nucleus falling apart, I'm going to draw it like this. So this is going to be like holes in it. So it degrades and it goes away. So then in the next phase, and I want to point out that as I'm talking about each of these phases, when the cell is undergoing mitosis, all of this just happens consecutively. Um, and so it just happens in one smooth motion. It's not like it stops, goes, stops, goes. And we come up with these steps just to help explain this process. Okay, so the next step is called prometaphase. Prometaphase. And in prometaphase, what happens is the centrosomes are now on the opposite sides of the cell. And from the centrosomes, these spindle fibers grow out of them, and each spindle fiber will attach to each chromosome. Okay, so I'm going to write a spindle fiber from the centrosome grabs each chromosome. Okay, so let me draw that. Okay, so here's my cell. There's no more nucleus, so the chromosomes are just floating around free. And the centrosomes are now on each side of the cell. So one's over here on this pole, this one's on the other pole. And I'm going to draw some spindle fibers in red just to make it look different. Okay, so there's 4 chromosomes. So 4 of these chromosomes are going to bind to the 4 chromosomes, the spindle fibers. So I'm going to draw 1 chromosome here, 1 here, 1 here, 1 here. Okay, and so remember we have 4 chromosomes, each one that's been replicated. And so this spindle fiber is also going to bind to each one. Okay, so this one's got to reach a little bit further because all the chromosomes are on the left side. And so the spindle fiber is going to grab the kinetochore on the centromere of each chromosome. Let's write that down. The fiber grabs, hold on, let me rewrite that. Grabs the kinetochore, that's the love handle, on the centromere of each chromosome. Okay, so that spindle fiber is grabbing that love handle that I mentioned earlier. Okay, then we're moving into the next phase. The next phase is called metaphase. I'm gonna scoot up. Okay, in metaphase, now that the spindles have grabbed each chromosome, they now can move the chromosomes wherever they want. And what they're going to do is they're going to line all the chromosomes up across the center of the cell, the equator of the cell, which is known as the metaphase plate. So the chromosomes will line up single file across the metaphase plate. Okay, and let me draw it so you can see what I'm talking about. And so this is going to be all controlled by those spindle fibers. So here is the cell, okay? And so we've got one centrosome here, one centrosome here, and they have the spindle fibers, and they're going to line up the chromosomes. Sorry, that's kind of messy. Okay, so let's say this is mom's number one, dad's number one, mom's number two, dad's number two. Okay, so these are your chromosomes and they all line up. And so I'm only showing 4, but remember with humans, we have 46. So it's going to be 46 lined up and all across the metaphase plate of your cells. That's a lot. And it's too much to draw because that would take all day. So I'm just going to draw 4. Okay, so now this is metaphase. Then we move on to the next phase. The next phase is called anaphase. And in anaphase, what happens is the spindle fibers are now going to separate the sister chromatids, pull apart the chromosomes, the replicated chromosomes. So in anaphase, the sister chromatids are separated. Okay, and this is what it looks like. Whoops, what's it doing? Okay. Okay, let me draw our cell again. So here's our cell. We have our centrosome. Here's our centrosome. Sorry, that's supposed to be 2 barrels. And so now we have these spindle fibers that are contracting. And then what happens is the 2 sister chromatids are pulled apart from each other. So here's number 1, number 1, number 2, number 2, number 1, number 1, number 2, number 2. Okay? And so that's what anaphase looks like. You could see the chromosomes being pulled apart. Okay, and then the last phase of mitosis is next, and that is called telophase. Oh my goodness, why is that not working? Oh my gosh. Sorry, my whiteboard all of a sudden isn't working. Okay, so this phase is telophase. And what happens in telophase is now all the chromosomes come back together and a nucleus is now going to form on both sides of the cell around the 2 groups of chromosomes. So in telophase, a new nucleus forms around both sets of chromosomes. Oops. Okay, and this is what it looks like. So let me draw our cell, okay? Oops, that's not a very good cell. And so in this cell, we have 4 chromosomes. Let me make them a little bit leaner. So here's one pair, here's another pair, and then over here we've got one pair, and then here's another pair. And then a new nucleus is going to form around each set. Okay, and so in our cells, we're going to have 23 pairs or 46 chromosomes. Okay, so while telophase is happening, this other thing is happening, which is a phase called cytokinesis. Okay, cytokinesis is not technically part of mitosis, it's a separate part of mitosis. So all of mitosis happens, and the cytokinesis happens at the end of mitosis. Okay, and so I'm just going to add this here, cytokinesis. And what cytokinesis means is the whole cell is going to start separating. Okay, so let me scoot up here. And so this is what that looks like. So as the nucleus is forming, the cell starts to form this cleavage in the plasma membrane. It looks like this, okay? And so, and the cleavage just continues to form until eventually the cell comes apart into 2 cells. Okay, so cytokinesis is the process of splitting the whole cell. So here are the 2 pairs or the 4 chromosomes. Okay, and so cytokinesis is when the whole cell splits into 2 identical cells. Okay, and so that's it, guys. That's all the details about mitosis. There is a lot. I want to pull in a picture of some real cells so you can just see what it really looks like. Let's see here. And so you can see what a real cell looks like when it's undergoing mitosis. And so, remember that mitosis is only happening. Oh no, I can't move it. Oh my goodness. For some reason, my whiteboard just stopped working. So it's very slow. Oh, come on. Okay, so you can see it here. Let me just do this so we can look at the picture. Okay, so here's the picture. I'm sorry it is like over my drawing, but that's because my whiteboard is not cooperating for some reason all of a sudden. So in the top left is prophase. And so remember, prophase is when the nucleus falls apart and all the chromosomes are just kind of all on top of each other. And then the next phase is prometaphase, which is not shown here because it doesn't look that different in the cell. Then we have metaphase, where the spindle fibers has attached to each chromosome and has lined up all the chromosomes across the middle of the cell. So the chromosomes are blue and the red are the spindle fibers, okay? And then the next phase is anaphase. This is where the sister chromatids are pulled apart from each other and pulled to the opposite sides of the cell. And then in telophase, the nucleus is reformed and cytokinesis also happens where the 2 cells are now going to split into 2 separate cells. Okay, guys, so that's it for today, and I will see you next time. Bye-bye.